Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with 12th Standard Biology Chapter 11, that is Biotechnology Principles and Processes. So in the last class, we have seen the principles of biotechnology. We have learnt about the recombinant DNA, origin of replication, cloning and plasmids. In today's video, we are going to learn about the tools of recombinant technology. So in this we are going to learn about the restriction enzymes, the recognition sequence, then the palindromes, what are palindromes, and also about the exonuclease and endonucleases. So let us begin. Biotechnology principles and processes, the tools of recombinant DNA technology. So what are the different tools and which are used in recombinant DNA technology? So the first we have are the restriction enzymes, then polymerase enzymes, ligases, vectors and host organisms. So these are the tools and this is the host organisms in which the modified DNA will be inserted. So first talking about the restriction enzymes. Now in 1963, two enzymes were discovered that could restrict the growth of bacteriophage in the E. coli bacteria. Now one enzyme added the methyl group to DNA. So the function of one enzyme was to add methyl group to DNA and the other enzyme could cut the DNA and was called as restriction endonuclease. Okay, so one enzyme added methyl group to DNA and the other one could cut the DNA. The first identified restriction endonuclease is the Hind2. This is the name of the restriction enzyme and it is Hind2. So the restriction enzymes comes under the nucleases, which is a larger group of or larger class of enzymes. Then now there are two types of nucleases. First one is exonuclease and second one is the endonucleases. So what does exonuclease do? It removes the nucleotides from the periphery or the ends of DNA. Suppose this is a DNA strand, the endonucleases will cut the peripheral part. So it removes the nucleotides from the periphery of the DNA, whereas the endonucleases which are there, it cuts the DNA at specific positions away from the periphery within the DNA. So the endonucleases can cut the DNA. It can cut the DNA at specific position within the DNA. Then talking about one example, that is the first restriction endonuclease, that is Hind2, which was discovered. It always cut the DNA molecules at a particular point by recognizing a specific sequence. So the enzymes could identify or recognize a specific sequence with 6 base pair. For example, we know that the A, that is adenine, always pairs with T, that is thymine, and G, guanine, always pairs with C, that is cytosine. So these were the base pairs. These are the base pairs and such a sequence was recognized by these restriction endonucleases and it was cut at only that particular sequence. So this specific base sequence is known as the recognition site for Hind2. Okay, so the Hind2 enzyme which was there, it could recognize a specific sequence of 6 base pair and this specific base sequence is called as the recognition sequence for Hind2. Now the restriction endonucleases cut DNA at specific position. Okay, this is the same thing. They cut DNA at specific position by recognizing the specific sequence of DNA, and that position is or that sequence is referred to as recognition sequence. Only that sequence the enzyme will recognize and it will cut them. Then each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. So what is a palindromic nucleotide? So a palindrome in DNA is a sequence of base pair that reads the same on two strands when orientation of reading is kept the same. So as we can see here, for example, the following sequence reads the same on two strands, that is 5' to 3' direction. This is also true if we read in the 3' to 5' direction. So let us read. Now this is a first sequence. We will read from 5' to 3' direction. So this direction we have G, A, T, T, C. So this is our first sequence. Then this is our second DNA strand, and from here also we will read from 5' to 3' direction, that is G, A, T, T, C. So we have the same sequence, that is the base pairs reads the same on the two strand when the orientation of reading is kept the same, meaning we are reading from 5' to 3'. Here also we are reading from 5' to 3'. So such a sequence is called palindromic sequence, and it will remain the same if we read from 3' to 5' also, if we read in this direction. So we have C, T, T, A, G. Here, from here also C, T, T, A, G. So now this is the palindromic sequence or the recognition sequence. Now upon identification of the recognition sequence, the restriction endonuclease or the restriction enzyme binds with the DNA and cuts each of the two strands of the DNA duplex helix at the specific point in their sugar phosphate backbone. So the restriction endonucleases or the restriction enzyme will cut the DNA, both the strands of DNA at specific points. So this is all in today's video. Hope you have understood my video. Thank you for watching. Please like, share and subscribe to the Bodhi Academy.